Thank you for the introduction, Bear. I hope that the content of my presentation today will give you deeper insights into the broadest spectrum of applications possible with the technological innovation behind the Synergy Neo2 system. So when we think about the drug discovery process, there are both mechanistic studies that are carried out to identify the mechanism of action of test molecules, but also quantitative imaging and microscopy to look at phenotypic and morphological changes that occur. Today, I want to focus on mechanistic studies and just, I would say, multi-mode detection modalities and applications that are relevant to our platforms as they relate to microplate readers. So we have a broad spectrum of microplate readers and imaging multi-mode readers as well that cover an enormous spectrum of applications and biological targets of interest. One category within our portfolio are hybrid multimode readers, which I will cover shortly, but that encompasses both quad monochromator optics and filter-based optics to drive a range of different complementary applications and assays. We have more, I would say, rudimentary, general workhorse microplate readers like the Synergy LX, and of course, our imaging multimode readers that has combined capabilities of microplate-based assays, so multimode detection, as well as quantitative imaging and microscopy. But of course, at the hierarchy of our microplate readers or multimode readers is our Synergy Neo2 system that has independent optical light paths, so it has quad monochromator optics and filter-based optics, which makes it a hybrid microplate or multimode reader. And it's fully featured and flexible from a perspective that it's modular and upgradable to allow you to add functionality to drive a broad spectrum of applications and workflows. Of course, the system is designed to cater to the most demanding needs of those users that require speed, sensitivity, dynamic range, performance for both biochemical-based assays and cell-based assays. Now, the modalities offered by the quad monochromator optical pathway on a Synergy Neo2 are those illustrated on the right-hand side of this slide. So UV-vis photometry, fluorescence intensity, which also includes FRET, as well as glow luminescence and flash luminescence. Now, with the quad monochromator system, because you have these different modes of detection, there's a huge arena of targets that we can detect, characterize, you taking a multidisciplinary approach as far as our labeling strategies are concerned. So here we are looking at examples of absorbance-based assays to look at enzyme kinetics, kinases, oxidative stress, apoptosis, so on and so forth, using different assay techniques, all of which are of course capable on the Synergy Neo2 and many others. Now, of course, the quad monochromator optical system caters exceptionally well to the realm of fluorescence intensity. And with the ability to dial into wavelengths without using optical filters, using a diffraction grating, we can select wavelengths of interest for excitation and emission and detect that biological or biochemical target. So one of the greatest benefits of our quad monochromator optical system is that it is a variable bandwidth mono as opposed to a fixed bandwidth monochromator, which is often seen in other commercially available systems. Now, with this capability, we offer users the flexibility of working across a broad spectral range of 250 to 900 nanometers and bandwidth adjustment between 3 to 50 nanometers in 1 nanometer increments. So what are the implications of this? Well, there are many applications where you have a fluorescent probe that has a low extinction coefficient, so its ability to emit light is compromised. In this scenario, one would increase the bandwidth at the desired excitation and emission wavelengths, resulting in a higher signal intensity level. Now, conversely, you may be faced with an assay scenario where the fluor exhibits a narrow Stokes shift, i.e. the absorption and emission maximas reside close to one another. When faced with this assay setup, you would constrict the bandwidths to reduce the amount of excitation light carryover and quenching of signal. Many fluorescence labels used in biochemical and/or cell-based assay constructs exhibit a narrow Stokes shift in the range of 8 to 15 nanometers. Here, we're looking at just a few examples of many fluorophores which present these characteristics. When detecting biological targets labeled with such fluors, it makes perfect sense to use a variable bandwidth monochromator to adjust the spectral wavelength and bandwidths to account for the spectral fingerprint of the label of interest. An example, one of many examples where this would be applicable, is if we're doing a FRET assay, fluorescence resonance energy transfer, using a CFP donor and a YFP acceptor. And you can see the spectral profile here. There's a lot of spectral overlap. And this is exactly a scenario where you would adjust the bandwidths for the excitation emission profiles of the donor and acceptor to make sure that there's no carryover of light, and then of course quenching of the signal. Now, as you can imagine, there are a huge arena of fluorescence intensity based assays out there. And here are just examples of some of those many different assay constructs that could be used to detect or characterize different biochemical targets. And our system gives you the full flexibility of looking at these and many other targets using different assay constructs, different assay approaches that are commercially available, or even assays that you would build yourself in a lab. Now, of course, luminescence is a very common technique to look at a number of biological responses. So cell viability, apoptosis, differential gene expression, and so on, especially in the field of cell biology. And here we can just see examples of different luminescence assay constructs used to look at those biological responses or changes. And of course, we have an independent light path that's used exclusively for luminescence detection on the Synergy Neo2 system. And it allows us to generate sensitive data with a wide dynamic range and in the most challenging of luminescence assay approaches. Now, when we are talking about our filter-based optical pathway, which is independent of the quad monochromator, we have top filter detection and bottom filter detection. Now, having the ability to use optical filters and dichroic mirrors earns you the ability to gain in sensitivity, higher transmittance of light, so therefore better sensitivity, and it opens up an even broader spectrum of modalities and associated applications, many of which are listed here or illustrated on the right-hand side of the slide. So filtered luminescence assays like BRET, which stands for bioluminescence resonance energy transfer, time-resolved fluorescence assays, both heterogeneous, homogeneous, fluorescence polarization, and of course, alpha screen. Now, with having the ability to detect signals from beneath the plate or below the plate using filter-based optics. This gives us superior luminescence detection capabilities, especially when we're dealing with cell-based assays. So if we're looking at cell viability using ATP as a reporter, or GFP transfected, looking at GFP transfected cells or cell migration, having the ability to have the detector in proximity to the point at which the cells are resident, so settled at the bottom of the plate, gives you much better performance characteristics and better sensitivity data. So many benefits of having bottom filter detection, aside from using other pathways that exist on the Synergy Neo2. Now, of course, all of our optical filters that contain, I would say, filter-based optics utilizes barcoded filter cubes that contain excitation, emission filters, and dichroic mirrors. And the fact that it's barcoded makes it so incredibly easy and straightforward to use in terms of positioning the correct filter cube and validating that the correct filters are used for the assay of choice when you're running an assay. So simplifies the entire process as opposed to guesswork and incorrect placement of filters. This is definitely the way to go in terms of labs where there are multiple assays, experiments, methods and workflows in place, and getting correct filter cube placements. And of course, we have a broad arena of filter cubes or barcoded filter cubes available to cater to a wide range of fluorescence intensity, luminescence, TRF, FP, and alpha screen-based assays. So let's talk about some of these enabling assays, such as filtered luminescence assays, using obviously our filtered optical light path. Here we're looking at examples of different BRET or bioluminescence resonance energy transfer assay constructs to essentially look at protein-protein interactions in live cells without having to lyse the cell membrane. It's a very, very powerful tool to look at cell biosensing and, as I said, protein-protein interactions. Here we exploring the dynamic protein interactions within cells using a protein-protein interaction system akin to BRET, known as nano-BRET, which is developed and promoted by Promega. A blue-shifted donor and red-shifted acceptor minimizes the background and minimizes, obviously, spectral overlap. Donor MDM2 and acceptor P53 fusion pairs, in this example, were investigated in HEC293 cells, assessing the binding inhibitor nuclein-3. IC50 values were obtained, which were obtained correlated well with obviously previously published data. Assay performance and Z factors between 0.5 and 1 clearly demonstrates low variability, as you can see in the table and graph on the right hand side of the slide. And so with this system, what you have is deeper insights into protein function with robust assay capabilities. Now, additional modalities that you inherit using the Agilent BioTek Synergy Neo2 system are alpha screen and time resolved fluorescence, or TRF. And just to give you a glimpse as to the range of biochemical targets that you can detect or study using these assay approaches is illustrated here in this table. Functional assays looking at GPCR signaling, receptor binding, a whole range of enzyme assays, interaction studies between proteins or protein peptide, protein DNA, as well as other functional assays where we're looking at cell proliferation or viability. There are a range of benefits of using these more advanced techniques like alpha screen or TRF over conventional fluorescence or absorbance, which I will cover in the next few minutes. And of course, because the Synergy Neo2 system is a modular system, you can see that one of the modules that can be configured with the Neo2 is a TRF laser. It's a 337 nanometer nitrogen laser, and it's optimally used in the context of driving homogeneous TRF-based assays. Examples would be HTRF or Lantha screen, and many others that have that kind of assay construct. Now, using the TRF laser, you have much better gains in sensitivity and speed, and that's clearly, and of course, dynamic range over using a xenon lamp, as you illustrated here by the table and the graph. But let's talk a little bit about the basic fundamentals of TRF. What is TRF? For those of you who haven't experienced or worked with TRF, let's talk a little bit about that for a few minutes. So time-resolved fluorescence is an assay technique dependent on using lanthanide chemistry. In the periodic table, there are a group of metals known as lanthanides, as we see here, and it's specifically samarium, europium, terbium, and dysprosium, which we use when we run TRF-based assays. The lanthanide in question comes in a chelated or cryptated cage structure, as you can see in the illustration on this slide, and it has a binding arm and chelate or cryptate associated with that lanthanide, which is then conjugated to your target of interest. So it could be a drug, a haptane, an oligo, a peptide, an antibody. And so there are different ways to label your target of interest. Now, why would we use TRF over conventional fluorescence or other techniques? Well, I think this data set speaks a lot to that topic. This was some data generated in the late 1990s looking at a chemokine receptor interaction, comparing conventional fluorescence labeling strategies like when using fluorescein, rhodamine, BODIPI versus using a lanthanide chelate such as europium. And you can clearly see from this graphical output that there's a significant leftward shift in the limit of detection. So it's a significantly more sensitive technique over conventional fluorescence. So you might be asking yourself, so why is lanthanide chemistry giving us better sensitivity data? Or why are we able to detect interactions or detect the concentration of a biochemical target to a greater degree than we would using a fluorescence label? Well, that's illustrated partly by this graphical representation. When we think of conventional fluorophores, some of those fluorophores exhibit a narrow Stokes shift. So the absorption maxima and the emission maxima reside very close to each other, like we see here with FTSE. However, with lanthanides like europium, samarium, terbium, dysprosium, they have a very, very large Stokes shift. So the excitation maxima is in the near UV part of the spectrum, so basically around 320-ish nanometers, and then your emission is far shifted towards the red end of the spectrum, around 615 or 665 nanometers. So the fact that there's no spectral overlap means that you are not quenching that signal and you're getting much better sensitivity data. Now, the other reason why TRF delivers better quality data, and I would say just general gains in sensitivity, really that's the crux of it, is based on the way in which the signal is emitted. Because of the emission of that fluorescent signal from the lanthanide, it's a much more prolonged emission profile, which means we can gate the signal, hence its time-resolved fluorescence. So we gate the signal, we exclude the timelines where we're getting high background or noise, and we basically gate the signal and we also use broadband filters that are separated from each other. And the basically benefit of taking this approach of gating the signal using large broadband filters means we have much better gains in sensitivity over conventional fluorescence or other techniques. Now, there are different formats of time-resolved fluorescence assays. There we have heterogeneous and homogeneous-based assays. A heterogeneous TRF assay is one where there are wash steps involved. So imagine you think of an ELISA assay where you immobilize the target and then you wash to remove unbound material. That is the same concept as what we do in a heterogeneous TRF assay. An example of that would be Delphia, so dissociation enhanced lanthanide fluorescence immunoassays. And then we have homogeneous or non-wash assays where we incubate the donor and acceptor pairs and we allow them to reach equilibrium or bind, and then we excite the donor, which is a lanthanide, and we detect the emission of the acceptor moiety, which is generally a fluorophore-like allopicocyanine, as we see in this example, or Cy5 or BODIPI TMR. Those would be the acceptor pairs used in a homogeneous TRF assay. Now, heterogeneous TRF assays are used in a broad spectrum of different assay approaches. So looking at enzyme assays, cytotoxicity, in some rare cases we see multiplexing, and then also functional assays and receptor binding. But overall, the gain of using time-resolved fluorescence-based assays is a gain in sensitivity, at least I would say 1 order of magnitude or 10-fold greater sensitivity over traditional fluorescence. We, minimize the risk of getting interference or autofluorescence from colored compounds, especially if we're, say, drug screening. We have the ability to miniaturize the assay into smaller volumes, so less reagents, less antibody usage because of the sensitivity of the assay technique that TRF offers. We can obviously multiplex to some degree using heterogeneous TRF assays, so being able to detect multiple targets in the same sample. And then, of course, the assay technique as a whole offers a much wider dynamic range versus traditional absorbance or fluorescence. So therefore, you negate the need to dilute your sample to make sure it falls within the linearity of the standard curve. Now, homogeneous TRF, which is the non-wash assay approach of TRF, allows you to do many other interesting or explore many other interesting avenues. So looking at protein-protein interactions, detecting the secretion of cytokines, chemokines, growth factors from stimulated cells, looking at protein modifications, cleavage, GPCR signaling, protein DNA interactions, and so on and so forth. So you can see the great degree of diversity and flexibility offered by these TRF-based assay approaches. Now, of course, another homogeneous, non-wash based assay approach, which I talked about earlier on, was alpha screen, which is one of the many modalities offered by the Agilent BioTek Synergy Neo2 system. Now, the alpha screen approach is one where we're using polystyrene beads, dextran coated, that are 200 nanometers in diameter, and they are conjugated to antibodies that recognize different epitopes of your target. So what we're looking at on the illustration of the left is just one example of how alpha screen chemistry is used. These beads are commercially available from various vendors, and it allows you to either buy unconjugated beads and conjugate them to your antibody of interest, or buy them pre-conjugated for the target of choice. So here we're looking at detecting a particular analyte. Could be, for example, IL-6, IL-8, or TNF-alpha. That's just one example, few examples. And then we have antibodies that recognize different epitopes of that target. Now, once you've incubated your sample with the beads in question, and you reach a point of equilibrium, you excite the donor beads with a 680 nanometer diode laser, and you get an energy transfer process, I would say a chemical energy transfer process, and the mediator is singular oxygen. Once that singular oxygen perfuses into the acceptor beads, you get a cascade of electron transfer steps and emission of light at a shorter wavelength than excitation, which means that you're actually getting much, much lower background levels and a huge gain in sensitivity. So gain in sensitivity, larger dynamic range, lower backgrounds are just some of the many advantages of using this alpha-based approach. Now, there are different assay constructs from the alpha world or alpha screen world. There are biochemical-based assays where we're detecting the binding between two partners, say a protein-peptide, protein-DNA, a protein-protein interaction. We could be working with the alpha LISA-based system, which is a brilliant alternative to ELISA. And in that context, the sample could be whole blood, could be serum, could be plasma, tissue homogenates, could be the supernatant from cell culture to detect different cytokines, chemokines, growth factors with much greater sensitivity gains than what you would see with ELISA and a much simpler workflow. And then, of course, we have the Alpha Screen Surefire assay, which is often used to look at phosphorylated cellular targets. Now, here we're really looking at the simplicity of performing an Alpha Screen assay. You know, an Alpha Screen assay is typically performed in between 2 and 4 steps. Addition steps may, of course, vary with the assay requirements. Now, the assay workflow is typically performed in less than an hour and a half to 2 hours. When you reach equilibrium after incubation of your donor and acceptor beads, and that obviously is dependent on the affinity of the binding partners, then you're able to basically excite the donor beads and detect the emission of light at a shorter wavelength from your acceptor beads. And the beads won't settle because they form they don't sediment in solution. They form a nice colloidal suspension, and because they're so incredibly small, 200 nanometers in size, they're not like large Cepheros beads, for example. And the reagents are incredibly stable. So many benefits in terms of the working mechanics of using Alpha Screen assay beads and the different range of biochemical targets that we can detect using this sort of assay approach. And as I mentioned to you earlier on, there are different bead formulations that are commercially available. Unconjugated bead, where you would conjugate them yourself, or pre-conjugated beads, donor beads, acceptor beads, with different sorts of labeling approaches. Now, for those who might potentially be interested in trying an alternative approach to ELISA as an immunoassay and using an assay that is non-wash, and you can run in 90 minutes to 2 hours with 2 or 3 reagent addition steps, the Alpha ELISA approach is definitely one worth considering. And there are commercially available kits available for different biochemical targets covering different disease areas. And many of these kits are based on human targets, murine, rat targets. And you can just see a rich tapestry of those here on this slide. Now, the other application I wanted to quickly cover off was fluorescence polarization. And that's a modality that you have on the Agilent BioTek Synergy Neo2 system. And FP is a powerful technique. It's a homogeneous-based assay workflow, so again, non-wash, where it's used to look at the detection of a small moiety to a larger binding partner. So, for example, a drug binding to a receptor, a peptide binding to a protein, an antigen binding to an antibody. And here are just examples of different targets that are studied using various FP assay approaches. Now, the technique is based on looking at polarization of emitted light, which is dependent on the molecular volume and, of course, rotational spin of the complex that's labeled. So if we have polarized light, that excitation polarized light that hits your I would say your target that's labeled, and its rotational spin is incredibly small because it's got a large molecular weight, so its rotational spin is slow, the light remains polarized as it emits. However, when you have a small tracer or a small moiety which has small molecular weight, faster rotational spin in solution, when you hit that biology, I would say, with polarized excitation light, the light becomes depolarized. So what we're doing in an FP assay is we're taking a small molecule, binding partner number 1, so that could be, for example, a drug, or it could be a peptide or an antigen, and it's labeled with a fluorescent tag, usually something like FITC. That's now known as your tracer, and when excited, it rotates rapidly during its fluorescence lifetime, and then, of course, the light is depolarized when it emits. When that tracer binds to a larger binding partner, its rotational spin slows down. It's bound to something much larger, and the overall complex increases in molecular mass, rotational spin slows down. And so when we hit that tracer with light, because its rotational spin has slowed down, there's a change in polarization of light. And that's what we're detecting using fluorescence polarization. How is that done from a mechanics standpoint? Well, we're using polarization filters in our bar-coded filter cubes, and we have parallel and perpendicular, or S&P, emission filters that looks at parallel perpendicular light that's emitted from that tracer. So that's how we're doing it in terms of using optical filters and dichroic mirrors. An example, or one of many examples where FP is used, is one where we're looking at the binding of a drug to a receptor, and here we're looking at a predictor, a Churg FP assay, which is often used in industry as a means of drug screening to look at the efficacy of a drug and its ability to bind to a target receptor. So all in all, now if we back out from all these wonderful applications that I've been covering with you, and we look at the hybrid optical system that we have with the Agilent BioTek Synergy Neo2, we can take a multidisciplinary approach. We can take different assay approaches depending on that target of interest. So let's say, for example, we're interested in studying kinases. Well, there are absorbance-based techniques, alpha screen, luminescence, time-resolved fluorescence, all of which have advantages and disadvantages of using those assay labeling approaches. And of course, when we're detecting absorbance or alpha or luminescence or TRF, we're using different optical pathways in our device to be able to detect that signal. But it gives the users the ability to select the ideal application or ideal method of choice for that target of interest, and not be locked into one particular technique. So the fact that the system has hybrid optics will earn you the ability to drive a varied approach to studying or investigating a particular pathway or target. Now, of course, you can imagine with hybrid optics, with all these different optical pathways and modalities that you can run on a Synergy Neo2, the range of applications is incredibly diverse and broad. And here, we're just looking at sampling of different applications and workflows that are easily run using our approach. Now, of course, there's a large degree of flexibility and, of course, modularity and upgradability with the Synergy Neo2 to add capabilities like a CO2-O2 gas controller and dual reagent injectors. Temperature control is a standard feature on all Synergy Neo2 optical systems. But of course, having the ability to add gas controllers and injectors opens up an even broader spectrum of applications that are dependent on modulating CO2. Let's say if you're doing a live cell assay or doing a hypoxia study, you would use the gas controller. You'll want to look at enzyme kinetics, metabolic activity, differential gene expression, ion channel studies, calcium kinetics. There are a whole range of applications where having the ability to add your rate limiting reagent and measure immediately after addition can be incredibly valuable in the context of that particular method or workflow. Now, many applications run over much longer durations, or we may want to automate them. So if you're doing a live cell assay, you can easily integrate a Synergy Neo2 system to our BioSpar, which is an automated incubator that can hold up to 8 vessels or 8 microplates and different types of vessels. We can automate an ELISA workflow where we have front-end liquid handling, to add primary antibody, secondary antibody, conjugate, stop solution, various assay buffers. We can do incubation steps in the BioSpar, and the back-end detection could be done on the Synergy Neo2. Or we could be running different assays, kinetically over time, or in parallel. So the flexibility to automate that workflow is incredibly valuable when integrating a Synergy Neo2 system with a BioSpar and or front-end liquid handling. So if throughput is a consideration for your lab, you have the ability to integrate the Synergy Neo2 to a BioStack Neo. And that allows you to load up to 10 or 30 or 50 plates. There are different stack columns available, and rapidly load plates to the Neo2 and back. So it's a bidirectional stacker, and it gives you incredibly fast plate transfer times. And when screening is of significance or importance, this is certainly the way to go. Now, what drives all of this technology and all these different applications and methods? Well, it's the power of using Gen 6 software, which is our next generation software, and it gives you all the tools, parameters that allows you to develop the methods, read and detect the chemistry of interest, and then carry out quantitative analysis, and then move on to the next step of your research or your workflow. There's an experimental hub on the user interface of Gen 6 software, which is incredibly intuitive. Everything is within a single plane. You have your navigation pane on the left. So it's a single navigation pane that shows you your method, your plate maps, your data views, your analysis parameters, exports, and many other useful tools that you will need access to. And then on the right, you have different views of the raw data, the calculated data, the exported data, and so on and so forth. Now, of course, there are lots of automation and data optimization tools or assay optimization tools built into Gen 6 software. So if we've got different volumes in each well, or we're going from one plate density to another, we're going potentially from 96 to 384 well plates, we can do probe height calibrations, we can do gain calibrations to adjust for different intensities of signal across the assay chemistry. And one of the greatest tools within the software user interface is the fluorophore spectra utility tool, which allows you to look at the spectral fingerprints of a large library of fluorophores and then basically define and set your bandwidth settings or your filter cube choices based on the spectral fingerprint of that fluorophore in question. And when it comes down to data analysis and exporting options, the choice is vast. Various quantitative analysis tools and different curve fit parameters, being able to determine IC50, EC50 calculations, Z prime calculations, and all of the critical analysis features that one would expect and hope to have on a single user interface when they're running all these biochemical and cell-based assays. And of course, for labs where 21 CFR Part 11 compliance is of significance, then we obviously have Gen 5 secure software, which caters to the needs of those users. So in essence, what we've developed here with the Agilent BioTek Synergy Neo2 platform is a multidisciplinary tool which offers hybrid optics, so the flexibility of working across a broad spectrum of wavelengths without using optical filters with the quad monochromators, but you also have high performance sensitivity using filter-based optics with barcoded filter cubes, using optimal excitation emission filters and dichroic mirrors, ultra-fast plate processing times, whether you're using a stacker or not. Its speed characteristics are definitely superior to many other readers that one might be even considering. And then, of course, the ability to run live cell assays, because there are modules that you can add like gas controller or injectors, and the ability to detect signals from below the plate using, for example, filter-based optics. And of course, an incredibly useful and intuitive user interface that allows you to capture the data, carry out the analysis, and then move on to exporting. So thank you very much for listening in on the talk, and I think now's a great time to answer any of the questions that you may have thought of as I've been going through my slides.
All right. Well, thank you so much, Charles, for that great presentation. Now we're going to get to some of the questions that came through. Let's see, our first one here is, when using the Neo2, am I able to build a protocol that uses both absorbance and fluorescence readouts in the same experiment from well to well?
Okay, great question. So I can think of a number of applications or workflows where that might be relevant. And simple answer to that is absolutely yes. So when we go back to what I was saying a little bit earlier on, we've got the experimental hub where we can build a protocol, and then we can add different read steps within that one protocol. So we can add, for example, the request here is, can we add an absorbance and fluorescence read step into that protocol? We can add multiple read steps, and then when we go into the read parameters for each of those, we can define what those parameters looks like. So for example, we could be doing an optical density 600 determination in absorbance mode, and then we could be doing a fluorescence readout because we've got a fluorescence tag or target in that same sample. Both done in the same experimental run in the same read, or two reads within the same run of the plate. So yeah, in short answer to that, yes, absolutely, yes. And it's even more flexible than that, but in simplicity, yes. Hopefully that answers your question.
Let's see, we have another one here. You described your barcoded filter cube system to be easy to use. With the microplate reader currently in our lab, we often face issues where the wrong filters were installed, creating confusion when time comes to read a batch of plates. Does your system notify the user which cubes to install when a protocol is selected?
Oh boy, the number of times I've come across that during my days as a research scientist and in many labs that I've visited in the past. So yes, that's one of the biggest benefits of having a barcoded filter cube construct. so that when you build your protocol and you come to run it, or let's just say even if you built it days or weeks prior, months prior, and you come to run it, if the correct filter cubes are not in place within the device, the software will prompt you, Gen 6 software will prompt you and say, hey, you need to have the following filter cube installed, and it'll tell you which filter cube has to be installed. And it's very, very, so it's basically a bulletproof strategy to make sure you're not using the incorrect cubes, and placement of the cubes is done in a very easy, simplistic manner where you just slide in the cube and it reads the barcode and you're done. There's no natural like processing or programming in the filter cubes before you run your assay. It's like incredibly straightforward and easy. So I hope that instills a little bit of confidence in whoever asked that question.
Great. Let's see. In your presentation, you mentioned that the Neo2 offers a dedicated luminescence light guide for detecting luminescence reporters. Is the light path I would use, is the light path I would use if I were to consider running the nanobret assay approach to the P-P interactions with dedicated filters? Also, can the Neo2 be configured with the optimal detector configuration for this assay setup?
Great question. So I think obviously what the person who's asking the question is referring to protein-protein interactions for other people on this call. I assume that's what we're talking about here, because that's where a nanobret assay is used in the context of. So if you're doing a nanobret assay, you would ideally, it's a filtered luminescence assay. So you would have to use the filtered optical pathway in the Synergy Neo2 system, not the dedicated light path that's only used for unfiltered lights. So if you're doing luciferase, luminol, just general visible light luminescence, that would be using the light path, the light guide. But if you're doing filtered luminescence assays like BRET1, BRET2, nanobret, all the different BRET constructs, you need to filter that luminescence light. So you would use the filter optical pathway of which is easily done. So you just have to make sure you use the right filter cube to detect the reporter used in that nanobret assay. And I think, sorry, that was part of the question about the configuration of the detectors.
Yeah.
Is that right?
Can the Neo2 be configured with the optimal detector configuration?
Yeah. So that's obviously, it seems like this individual knows that assay really well and the significance of having the right detector. I'm glad they brought that up, actually. So when you're doing a filtered luminescence assay, especially a BRET type assay like nanobret, you want to make sure that you're using a low noise PMT detector. So you don't want any interference with high backgrounds of luminescence light. So a blue shifted detector is one way of thinking of it, or a low noise PMT detector. So the nice thing with the Agilent BioTek Synergy Neo2 is you can configure it with the right type of PMT when you're building and designing the system. When you come to invest in the system, you could configure it with one low noise detector or two low noise detectors, as opposed to a red shifted detector, which would be looking at light, above 700 nanometers, so in the 850 range. So in short answer to your question, yes, absolutely. And it is very configurable. So hopefully that answers your question.
Perfect. Yeah, thank you. Let's see, we have another one here. It appears from your presentation that the Alpha ELISA assay system offers several advantages over absorbance ELISA. How does the cost per assay point compare to a traditional ELISA kit? Also, could you please confirm again if the assay workflow involves 3 steps, including incubations and runs in under 2 hours without washing the immobilized target?
Okay, yeah. So it's nice to see that some folks on the call are familiar with the Alpha ELISA system, or to some degree, I guess. But so yes, let's start with the cost per assay point. So if I think about traditional, I guess, suppliers of ELISA assay kits, there's so many. One of which just comes to mind instinctively is R&D Systems. So they have kits for different cytokines, chemokines, growth factors. So let's just say if you're shooting for IL-6 or TNF-alpha, IL-8, you want to get a 500 assay point kit. It will probably cost in the region of around, I don't want to quote exact numbers, but it's probably in the region of 500 to 600 US dollars for 500 assay points. An alpha Leiser assay system, which you can source from different suppliers like BioSystems, they're a great supplier of this assay type, or Revity is another one. It will probably be a very similar price. Maybe there is a slight cost saving, but the real advantage is the efficiency of that workflow, which was another part of that question. So instead of running an ELISA assay system that spans over many hours with wash interventions, here we're mix and measuring, adding acceptor beads, incubating for 30 minutes, 1 hour, then donor bead, incubating 30 minutes, 1 hour. So it's 2 steps of incubations followed by a read step. So yeah, it's a much more simplistic workflow with only a few reagent addition steps, 2 incubation steps, and a read. So that means you run the assay in 90 minutes to 2 hours, let's say, is the total run time of running that assay. So workflow efficiency, but the real gains are, I would say, workflow efficiency, sensitivity, linear dynamic range being much, much larger than what, say, a general ELISA assay would offer. Yeah, I think that answers the question. I assume that covers the aspects of the question that was being asked.
I think so. Thank you. Let's see, we have a couple more here. How modular and upgradable is the Neo2 system? We are an academic lab with a limited budget to work with and are currently in need of a flexible microplate reader, but will likely need to add on options in the future as the need of our labs change.
Reminds me of my days as a postdoc being in an academic lab with limited funds for investment into capital equipment. So yeah, so one of the nice things with the Synergy Neo2, among many other platforms in our portfolio, is the modularity and upgradability. So I think the question is about what's the degree of modularity or upgradability. So you could start with a Neo2 system that has, say, absorbance monochromators with single PMT, single filter cube filter optical pathway, and then add additional components down the road. Those additional components can vary from injectors, gas control, a dual PMT, filter cube light path construct, alpha screen laser, stackers, barcode readers, bottom filter cube optics. So there are a number of upgrade paths that are available. These are all done in the field, so we're not having to remove your system out of your lab. We have it done in the field. Many of those upgrades are done by the user, and then the ones that require much more technical intervention, we have a service technician or engineer would come to your site and do that upgrade for you in your lab. So your downtime is going to be minimal. So sorry, was that the question? So it's basically how much modularity and upgradability,
yes.
Is that right? Yeah.
Yep,
okay.
exactly.
So hopefully that addresses it for you.
All right, we have 2 more here, or time for a couple more. Do you have any examples to share of using your system to look at small molecule screening?
Small molecule screening. Many examples, actually. So our applications lab in Vermont has done quite a bit of stuff in this realm with many of our devices, but even with the Synergy Neo2. One that comes to mind is the BRD4 bromodomain TR fret assay. It's a competitive assay construct, and it's designed for small molecule inhibitor screening. And in that assay system, the BRD4 bromodomain is labeled with europium chelate, and that's your donor, and biotilated peptide containing the acetylated lysine serves as the ligand. And then APC labeled avidin binds to the peptide substrate via the biotin moiety, and that's your acceptor partner. And then when you have the donor and the acceptor in close proximity to one another, you get this resonance energy transfer process, light energy transfer process from the europium donor to the APC acceptor. So that assay definitely comes to mind. And that assay is traditionally run in low volume 3D4 well plates where cells are added to the well, and that assay is run immediately without you having to wait for the cells to settle. So the cells are in suspension and you're running the assay. And it's a very convenient way of doing it. And I think the folks in our lab who ran that assay and have published on it, used a compound library from Screenwell Natural Products. It's a Screenwell Natural Product library from Enzo, if I remember correctly. It's about 500 compounds. And they did a screen and they saw pretty much no inhibition with most of the compounds. And then a subset of those compounds showed something like 20 to 30% inhibition. And then there was a very, very small population of compounds that inhibited beyond like 30 or 35%. So we've got lots of good data, and that's just one assay type. But there's a whole range of different assay constructs that are not even TR-FRET based that could be used in that type of arena. Sorry, that was a bit long-winded, but hopefully it answers the question in relation to that.
All right. Well, looks like we're at the top of the hour. So that is unfortunately all the time that we have. I just want to thank you again, Charles, for a great presentation. And on behalf of Agilent, I want to thank everyone that joined us today. If we did not get to your question, we will follow up with you via email. And if you would like more information about Agilent BioTek's products and services, including the archive of previous webinars, please visit us at www.agilent.com. We hope that you enjoyed today's presentation and have a great rest of your day. Thank you.
Thanks, Bear. Thanks, everyone.